Dear students, now in the topic translation, I am going to explain you Wobble hypothesis. What do you mean by wobble? So wobble means you might have played with the wobbling doll in your childhood. So it is you will find it is keep on there is a unsteady rocking you will find. So that is the wobbling. So it is the Francis Crick in 1965 proposed the wobble hypothesis to explain the degeneracy of genetic code or codons. So why many codons to translate in a single amino acid? You will find there is a redundancy, there is a synonyms. You will find a single amino acid, they are specified by many codons. So why? There are 61 triplet codons coding 20 amino acids. That means to recognize these 20 amino acids and 61 triplet codon, you may expect 61 tRNA types are required for base pairing because they base pair with the mRNA codons. So that's why you will expect the similar 61 anticodons to recognize 61 triplet codons. But actually there is a 40 to 60 types of tRNA in the cell, or else it is less than that. Sometimes it may be 30 to 40, or else even 22 tRNA you will find in the human mitochondria. So then how they can able to recognize 61 different types of triplet codons, that is those are present on the mRNA. So how 22 tRNA or 30 tRNA can recognize 61 different triplet codon, because they form a complementary base pairing. For example, GUC for valine, CAG for it is for glutamine. So you will find, so this how this can be read by 30 tRNA. So this is what the wobble hypothesis is going to explain you. According to Francis Crick, wobble hypothesis, only the first two bases of the codon have a precise base pairing with the bases of the anticodon, while the pairing between the third base of the codon and the anticodon may wobble or move unsteadily. So this is what you can see, the base pairing with respect to, you can see the first two, the you will find the bases of the triplet codon base precisely correctly. For example, the first these two base pairing is typical base pairing, complementary base pairing. With respect to the third base of the mRNA and the first base of the tRNA, so they wobble. It is not the strict base pairing, or what you see the complementary base pairing or Watson and Crick base pairing, it is also called. So complementary base pairing between the codon of mRNA and anticodon of tRNA is less specific with respect to third base of the triplet codon and the first base of the anticodon. So codon will be recognized from 5 prime to 3 prime. So this will be first, second, third. Similarly, the tRNA molecule, this is the 5 prime to this is the 3 prime, it is carrying. So this will be the first base. So see here, this is the leucine, this tRNA is carrying leucine. So it will going to form a base pairing with the UUA, that is coding for leucine. So here in this mRNA there is a code UUG. The same tRNA can make a base pairing with this, because so it can base pair the A pair with the U, A pair with U, whereas here U pair with the G. So this is less stringent or not specific. At the first you will find in the first base of the anticodon with respect to the third base of the the triplet codon. So there is a strict base pairing rule applied only for first two bases of the triplet codon, while the first two bases for this, the strict base pairing will be, you will find in this, not with respect to this. So you will find the third base codon appears to be less important. This is known as Wobble hypothesis. So here you will find this foot is firm. Similarly, the first two bases proper strict base pairing, whereas the third base is wobbling like this, the doll. So it is pairing, G pairs with the C here, G pairs with the U here. So this is what you can see, wobbling base pairing. You can see the third base pairing is not very stable, it wobbles. Here in this you can see the CU, CUG, CUC, CUA. These codons represent the same amino acid leucine. So the first two bases of the codon form a strong base pairing. You will find here GA pairs with CU. Okay, but whereas the third one, the base pairing is not, you will find strong base pairing, you will find G pairs with the U. So this is called wobbling base pairing. So wobble is the term used to refer to the fact the non-Watson-Crick base pairing. So this is a perfect base pairing, what you see. Here these are the wobbling, C pairs with the A pairs with the A, G pairs with the, all these they are carrying valine. Okay, so only the perfect base pairing here you will find. Only first two, the base pairing is perfect, GU you will find. Where with respect to the third base, you will find the wobbling base pairing you will find. So this is what you can see. This wobble position allows some tRNA to pair with the 2 or 3 codons, thereby reducing the number of tRNA required for translation. So if this is a perfect pairing with respect to all codons, you will expect 61 triplet codons and 61 tRNA. Whereas 32 tRNA means there may be this type of base pairing. So you will find the base pairing is not stringent or perfect with respect to this base pairing. So that is how it is called not a regular Watson and Crick base pairing. So the base pairing is the wobble pairing, it is called. So at third position, unusual base pairing, which is against the base pairing rule. Adenine, cytosine, uracil may pair with A pair with I, C pair with I, U pair with I. So what is this I? So I stands for inosine. There is another, you will find in the tRNA a unusual base you will find in the first position of the anticodon, that is called inosine. So what is inosine? Is a modified adenosine. You will find the adenosine by catalyzed into the inosine by deamination process. There is an amino group in this position that is removed by the adenosine deaminase enzyme. So that gives the inosine. Inosine is also a purine base, and you will find in the anticodon position in the 34 base, you will find in the tRNA from the 5th region, if you count in the 34 position, there is a inosine. So this inosine can pair with U, it can pair with C, it can pair with A. So all these carrying alanine, So GCU, GCC, GCA, all these the coding for the amino acid alanine. But the same tRNA carrying, recognizing all these different triplet codons you will find. So this is what you can see. So this inosine is a modified adenosine in RNA generated by deamination of the adenosine in a process called this is called adenosines to inosine RNA editing. These are the post transcriptional modification of the tRNA. You will find all these unusual bases you will find in the tRNA, pseudouridine, dihydroxyuridine. So similarly there is an inosine you will find in the anticodon region. So this inosine can pair with the A or C or with U. So this is a post transcriptional modification. So that's why the position 34 inosine may base pair with UCA, you will find here. So Wobble rules. So relaxed base pairing allows the anticodon of the single form of tRNA to pair with more than one triplet of mRNA. So tRNA anticodon first base U can recognize A or G. So you will find the anticodon, you can see the U can recognize A or G. First base G can recognize U or C. So you will find here in the table the anticodon in the tRNA, the first anticodon in the tRNA, that is inosine, it can recognize the third prime position of the mRNA A, U, C. It can pair with A or U or C. So U of the anticodon, that is in the first position of the anticodon, it can able to pair with the third position of the triplet codon A or G. Similarly, G can pair with U, C or U. C can pair with G. A can pair with only U. So this is what you can see. This is mRNA, CCU, CCA, CCC, CCU. All these are coding for proline. See here the tRNA. So see GGI, 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 GGG. So GGI bringing, they are the carrying proline, and they can recognize, they base pairing with all these three different mRNA triplet codon. Okay, so this is what you can see, the Wobble rules. So tRNA is a molecule, it is an adapter molecule. So it is an adapter, the adapter, how you are using electrical device adapter or else your charger. So it has, you can see the adapting the current with that of your mobile. So adapter, so molecule. So it is adapting between the adapter between the mRNA and the polypeptide. One side it is it is carrying, it is the physical adapter with respect to the amino acid, and also informational adapter with respect to the mRNA. So this there is a isoacceptor, you can see it is a isoacceptor tRNA that have a different anticodons but carry the same amino acid. So here the anticodon UUG carrying the glutamine. Similarly, anticodon CUG carrying the glutamine. These are called isoacceptor, you will find. So what are the significances of this Wobble hypothesis? So the several codons for the same amino acids are recognized by the same tRNA. In this way, the minimum 32 tRNAs are required to translate 61 codons on mRNA. So 61 codons, triplet codons recognized by 32 tRNAs. So for example, the serine, 6 mRNA codons, there are 6 triplet codons specify serine, but only the 3 tRNA can able to carry the serine. So this is because of Wobble base pairing. Then position 34 inosine, this is the inosine that is in the 34 base position. So this base pair with the uracil or else C or A, inosine at the Wobble position to promote the reading of multiple codons are essential for the survival. So it can recognize multiple codons. So that is what is the advantage of that? The ribosome has a mechanism to place the check with the first and the second codon bases are complementary to the third and the second anticodon. So in the mRNA, they can able to check these base pairing, these two base pairing, not with this. It has not a mechanism to check with the base pairing with that. So what happens, you can see third codon base on the mRNA and the first anticodon base of the tRNA will match anyway because of this Wobble pairing. So due to the Wobble base, there is a lesser chance of mistake during translation. Example, leucine codon CUU was misread by the CUA, CUG or CUC during transcription. Still, translation leucine, because all these are coding for leucine. So this is what you can see. The tRNA can you can see the recognize any of this the triplet codon, the same you will find here also. So the leucine here, GAG, a perfect base pairing with the CUC, but it also CUU also coding for leucine. This GAG also recognize this. So this thereby the tRNA number will be reduced, as well as the chances of misread will be avoided by this mechanism. So protein synthesis is protected against mutation of a single base. So the third position, so it is always, you can see, the third position base of triplet codon get mutated so many times. If it is get mutated, for example, U is replaced by A, so this for example GUU mutates into GUC, GUA, GUG, will be still be translated valine only, because the this is what you can see, the it will be recognized by the tRNA. It is same tRNA will recognize. So thereby the mutation is not going to be get translated. So this is also thermodynamic stability of Wobble base pair is similar to Watson and Crick base pair. So even though it is called Wobble pairing, the base pairing will be thermodynamically stable. So Wobble allows faster dissociation of tRNA from mRNA and also after protein synthesis. So these are all the, so this is the protein synthetic machinery. So where you will see the tRNA can make a Wobble base pairing with the mRNA triplet codon. So thereby it reduces the number of tRNA required for the protein synthesis. This is about Wobble hypothesis.